To get started, yeah? Okay. Thank you, everyone. A very good afternoon. Thanks for coming for, I think, the last talk of the year, the Armor series. Before I start and introduce, I want to acknowledge that the Field Museum resides on the ancestral homelands of the Three Fires Confederacy, the Ojibwe, Dawa, and Potawatomi. The area was also a site of trade, gathering, and healing for more than a dozen other Native tribes. My respect and gratitude to the many Native people who live here today, as well as their ancestors. The land we walk on was and remains Native land. Dr. Bryan Stuart was a PhD here in the Field Museum in a joint program between the Field Museum and the University of Illinois at Chicago during the years 2001 and 6. During that time, he did extensive field work and deposited herpetology collections here in the Field Museum from Laos, Cambodia, Vietnam, and Thailand. And in the world, the Field Museum has one of the largest and most important collection of reptiles and amphibians from Southeast Asia. And Bryan played a significant role in making us achieve this status. At that time, his herb collections were the largest anywhere from Laos and Cambodia in terms of the taxonomic and geographic breadth. And from 2006 and 2008, he was a postdoctoral researcher at the Museum of Vertebrate Zoology, supported by his MacArthur Foundation grant with Harold Voris and Bob Inger, run through the Field Museum. In 2008, he became the research curator at herpetology at the North Carolina Museum of Natural Sciences, where he remains today. And he continues to make extensive use of his collection in the Field Museum as a research associate. And thank you so much, Bryan, for making the travel to come and speak to us. Before I give you the floor, I also want to acknowledge the IT folks crew who always makes this possible, and Todd for organizing all the logistics. Bryan, the floor is yours.
Thank you so much. Thank you so much for having me here. I'm so pleased to come back home to the Field Museum. I have to be honest with you, the last couple of weeks have not been easy. And this trip home to the Field Museum in Chicago has given me something to look forward to. And I thank you very much. I want to tell you today the story, of course, of the Lao Newt. It's a story of discovery. It's a story of description. It's a story of, I think, surprise. It's a story of sadness. And it's a story that sort of ends, I think, with hope. And while I tell you that story, I'm hoping that I'll get across to you a subtext. And that subtext is actually what a profound and lasting positive impact the Field Museum has had on my career, and also on the career of my collaborators in Southeast Asia, particularly one person, Somputwan Pimachak from Laos, who I'm going to talk about extensively today. But before I go any further, I just want to introduce you a little bit to my museum. I'm at the North Carolina Museum of Natural Sciences. The main building that's open to the public is in downtown Raleigh. It's two buildings connected together by a glass bridge. And that big globe, that's a three-story theater where we can project all over the inside of it and give talks and so forth. Have any of you ever been to the North Carolina Museum of Natural Sciences? I see a few hands. Okay, wonderful. We are the State Museum of North Carolina. We are not part of North Carolina State University, although we have very, very close connections. Many of us are adjunct at North Carolina State University. We even have some joint positions, meaning some positions that are 50% university, 50% museum. But we are in a separate institution. We are the state of North Carolina. Our museum is free of charge. General admission is free of charge, and we get about 1.2 million visitors a year. And that makes us tying with the Biltmore Estate in Asheville as the #1 most visited tourist destination in the state. We happen to be located right across the street from the state legislature, which appropriates 80% of our budget, and I think that's very advantageous to us. It means we can constantly share with our legislators what we've got going on at the museum. And perhaps more importantly, we provide them with a great venue for lunch every day. And that really helps. But this is a museum I think you should know, because one might also call it the Field Museum of Natural History South Campus. Why do I say that? Because there are so many of us at the North Carolina Museum of Natural Sciences who came out of the Field Museum. So for example, Paul Brinkman, I hope you know Paul. Paul is holding a book he just published about 2 months ago. It's on the Elliott and Akeley 1896 African expedition. It's for sale in the Field Museum's gift store. I hope that you'll read it. It's a terrific book. He's our historian of science. He was a preparator here. He helped prepare Sioux for many, many years ago. Lindsay Zano is our head of paleontology. She was a postdoc here. And Lisa Herzog was one of our preparators here, and she is now in charge of paleontology operations at the museum. And I think many of you know Brian O'Shea. Brian O'Shea is our collection manager of ornithology. And Brian, correct me if anyone knows if I have this wrong, but Brian was working here in the bird collections, I believe, since he was a high school student, learning to prepare specimens and so forth. So it isn't just me who has benefited so much from the Field Museum, but others, my colleagues. And that's why I call it the Field Museum South Campus sometimes, if you'll allow me. But anyway, just very quickly, one of the things we take a lot of pride in at our exhibits are on, many of our laboratories are behind glass, so they can be viewed, interacted with, much in the way that you do with the Pritzker DNA lab here and also your fossil preparation lab. We do similar things with our paleo lab, molecular lab, biodiversity lab, and so forth. We have these little things called investigate labs that are adjacent to them, where members of the public can come in and actually participate in activities that are relevant to the research lab that's next door. For example, this child who's learning to extract DNA from a strawberry and so forth. And one of the things I just want to point out, one of the things that's unusual about the North Carolina Museum of Natural Sciences, I think, is that we have a very large living collection. And somewhat in the vein of the California Academy of Sciences, if you've ever been there, where we have a large collection. We have a whole department called Living Collections, and we have 2 full-time veterinarians on staff. And so we have all these live animals, which is very heavily amphibians, reptiles, and fishes, are incorporated into our exhibitry, our public programs, and so forth. What you're seeing there is a colleague of mine from China. She studies Chinese giant salamanders. Here she's looking at their North American relative, native to North Carolina. That's a hellbender salamander, which is she's seeing one alive for the first time. The sister group to what she works on back in China, and we have a living conservatory with butterflies and all sorts of things like that. So I just wanted to point that out. I'm not involved in the living collections. I'm the curator in charge of the research collections, but because it's so herp-centric, our exhibitry and our programming, because herps do well in those kinds of scenarios, they often pull us in for content. And I just want to close up introducing my museum by showing this building here, which is a very unremarkable looking building on the outside. We often say it looks like the backside of a Kmart. Is actually our fluid collection facility, and that's where I am, and it's about 6.5 miles away from the downtown museum that has the public exhibits that we were just talking about. This is closed to the public, but it sits on 44 acres of land where our education department has a whole nature trail set up around it. It's called Prairie Ridge Eco Station. That sort of talks about how one might have a sustainable, wildlife-friendly, native plant sort of environment in an urban, suburban setting. But anyway, that collection facility is specially designed for housing our fluid collections. So herpetology, of course, is out there, fishes is out there, and our invertebrates are out there. And we have all of those collections are out there of what I just named. So meaning the dry skins and skeletons and so forth for herpetology or fishes or whatever are out there as well. We keep our collections together. But anyway, so all the curators and collection managers and technicians and so forth are out there. This is what the herpetology collection looks like. The herpetology, we have a lab, there's a types cabinet. Anyway, just to look at our ultra cold freezers. And the 4 of us standing there, what's unusual too about the herpetology unit, where I'm now the curator, is that we have such a long living institutional memory. We have 2 curator emeritis One, Bill Palmer on the far left just passed away, unfortunately. But just up until a couple of years ago, I had actively working with me one emeritus curator who was there since 1961, one who's Alvin Braswell, who's still active. He's been there since 1968. My collection manager next to me in the blue shirt, he'll have been there 40 years next June. And so I tell you this because it's a little bit unusual. I come in with a lot, you know, I'm still the new kid there, and I've been there since 2008. And of course, we benefit, like you all do, from our wonderful technicians and interns and volunteers and so forth. Since I've been there, we've been very busy trying to capture all the data from our catalog ledgers, get them into our online database. Very quick plug, we're moving over to Specify, and we currently have advertised a position for a collection database manager, museum-wide collection database manager. It closes on December 5th. If you know of a good person, we would love to have them. So let me know if that might be the case. Somebody who can help us move from what we currently have into Specify. And then I've made big efforts since I've been there in all those years of trying to build up our genetic resources collections. We're not on liquid nitrogen, we need to be, but we've come a long way. When I arrived in 2008, in herpetology, we had 11 tissues and they were in a refrigerator. And today we have, well, as of this morning, we have 11,206 vouchered tissues, vouchered specimens with tissues. So it's changing quickly. And then this is the end of what I wanted to show you at my museum. This is my office filing cabinet. What would FMNH do is a sticker that I have on my filing cabinet. And why do I have that there? Because the Field Museum, I have a hard time as a museum curator of not seeing my job through the lens of what I developed while a student here. This was my very first trip to the Field Museum. This was in May of 1999. So this is before I was a graduate student. I was working with the Wildlife Conservation Society, the Laos program and also the Cambodia program. I was doing amphibian and reptile surveys around the country. And I was trying to identify this stuff that I was collecting, and I set up a relationship. I had the great pleasure of getting to work with Harold Voris and also Bob Inger. They were helping me with those identifications. I started collecting specimens. I learned what the value of specimens are, sent them back to the Field Museum to get identifications. And that's how this relationship started. And then, and I started doing that actually in 1998. In 1999, I came back to start to look at some of those specimens. And so, yeah, this is before I was a student. While I was working with the Wildlife Conservation Society, I was benefiting tremendously from the Southeast Asian herpetology collections here, and especially from Bob Inger and Harold Voris. And I just want to show you, that's Fouillon down there. Yeah, that's Fouillon. And there's Alan Resitar. Check out his computer. And that's what the collections look like when they're in that main hallway. Anyway, I look exactly the same as I did in May of 1999, so I don't need to point myself out. That's me between Bob and Harold. Anyway, I just described that relationship. It's been a wonderful journey. I've been working in Laos, like I mentioned, since 1998 and continue to work there and to today. And Laos, the Southeast Asian, the landlocked Southeast Asian country of Laos is sort of the focus of what I want to share with you today. This is just a mosaic of new species I've described to science, okay? I've described, I don't know, it's around 100 now, right? And it's so, just like so many of you, okay? The reason I'm mentioning that is not to boast numbers or anything like that, but to tell you that of all these species that I have described new to science since I began this venture back in 1998, well, I just want to mention, and this is probably a slide I don't even need to impress upon you, but I think there's tremendous critical importance in trying to recognize diversity and describe species simply because of the very dire conservation issues that are going on in Southeast Asia. Relatively the most rapid rates of deforestation, tremendous endemic biodiversity, also serious issues with wildlife trafficking and trade, and that comes into the story here later. But in any case, so I think that this exercise of naming, describing species is critically important for conservation. But this is what I want to say to you. After all these years, 25 years or whatever it's been of naming species, you know, today I'm still working on the very first species I ever described to science. And it's a difficult story, and that's what I want to share with you today. In 1999, I had the opportunity to get into an area, a difficult area to work in Laos. Even it's not a very heavily forested area, up in Saisumbuon province, what is today Saisumbuon province. Saisumbuon province, during the Vietnam War, Long Chang it was called, or Spook Heaven. At one point, so that is where the US CIA had their headquarters. And at one point, there were up to about 30,000 CIA agents living in Saisumbuon. This is where the whole Air America program, which was the bombing campaign from over Vietnam, was operated from. In that town, where 20-something thousand or more CIA agents were running around, was living in these small streams at high elevation, was this salamander. And I brought some specimens back to the Field Museum, and on that May 1999 trip, I got to look at them and compare them with some other things. And myself and Ted Pappenfuss at Berkeley, who at the time was working extensively on Asian salamandrids, we described it as a new species of science in the genus Paramesotritton, based on these few specimens I brought back from northern Laos. And it was a really remarkable looking thing. Most of the salamanders in this group, they don't have any dorsal coloration. Most of them are black, brown, not very colorful on their backs. A lot of them have those colorful bellies. And this thing is big, and it's warty, and it's a really remarkable looking salamander. We placed it in the genus Paramesotritton. We weren't totally sure. Didn't take long for people to generate molecular data and show that, no, this thing is not a Paramesotritton, but basically what it is it's either the sister taxon to or fairly basal within this clade of Asian salamanders, Paramesotritton, Pachytritton, and Synops, okay? But what we know, and it still jumps around a little bit. We've got a paper in review looking at genomic-wide sequencing data, and I can just tell you that this thing is a very basal lineage within these Pachytritton, Synops, Paramesotritton thing. It's been since named as a monotypic genus. Whatever it is, it's very, very old. It's perhaps 14 million or more years in age. That there it sat, and this is a map. This is a map. Did you know that Laos is the most heavily bombed country in the world? The United States, we dropped 50% more bombs on the country of Laos than we did on the country we were at war with, which was Vietnam. 2 million tons of bombs were dropped on Laos between about 1963 and 1974. And where that arrow points, so what this map is this is sort of orange indicates the most heavily bombarded areas. By the way, when you do field work in Laos, you see, that for example, is at the bottom is a bomb crater pond. These bombs would go off, they'd make a crater. Today they're filled with water. My colleagues and I did a little study one time looking at amphibian diversity in bomb crater ponds. A little bit dark and ironic, but nonetheless here, and in the forest, sometimes you still see lots of unexploded ordnance, which continues to cause injuries and casualties to the local residents when they accidentally stumble upon them, usually doing agriculture. In any case, where that arrow points to, that's the type locality of the Lao Newt. And so all I wanted to impress upon you is that there's this very, very old lineage of this remarkable looking salamander sitting there in this spot in northern Laos. And I guess we did all the best we could, but we failed to bomb it out of existence, okay? It got through that. And this, by the way, as you'll learn in just a second, is about the total global range of the Lao Newt, okay? And then this happened. So here I was, a new graduate student, and I published my first paper. It was a description of a new salamander from Laos, and I published it in the Journal of Herpetology, that 2002 paper I showed you. And I wasn't even working on salamanders for my PhD. I was busy. I had frog work to do. And so I moved on, like we all have done, right? You've described your species and you move on to the next. I moved on, and then something unexpected happened. It wasn't only me and Ted and the people who were interested in Asian salamanders from a taxonomic or phylogenetic perspective noticed that this salamander is unusually colorful. It's very large and warty. It's a weird thing, and it comes from a remote, hard to get to, bombed out piece of the world. And people decided When we published that thing as a new species of science in the Journal of Herpetology, people decided they wanted to have it as a pet. That is what happened. And so commercial collectors, initially from Europe and Japan, used the map in the original species description as a roadmap to get to the type locality. In fact, there was even a photocopy of that Journal of Herpetology paper once found in a remote village in that area where somebody had brought it and said, this is what we want, okay? That journal, it's not, people just heard about this thing. I mean, the Journal of Herpetology publication was used to go there and collect it. And there's something very unfortunate about this salamander. Again, a great irony. I just told you that this thing survived having its entire global range within the most heavily, one of the most heavily bombed areas of the most heavily bombed country on the planet. It survived through that. And yet, ironically, this little war salamander, to its detriment, we now know, is a peace lover. So unlike those other members of those other Asian newts, those Paramesotriton, those Pachytriton, those Cynops, those things are hard to keep in captivity. They're really aggressive. They tear each other up. They bite each other. They're very territorial. Not the Lao newt. Unfortunately, the Lao newt has some biological attributes that have contributed to its demise. And that is that, one, it is active during the day. They congregate in these stream pools. And as you'll see in a second, they're not distributed throughout streams. They're just at the high elevations of certain streams. And they congregate together in numbers, together in these pools. They don't fight. They get along great. So they're easy to find. In a matter of minutes, you can scoop them all out of that pool, and you can store them together in buckets. And they'll live together in an aquarium and so forth until they make their way around the world into an aquarium. So unfortunately, those attributes of being diurnal and congregating together and getting along peacefully in that war zone has made them very desirable in the pet trade, very desirable. And so after this happened, I was horrified. Here I was a graduate student. I was just trying to help conserve the thing, right? Isn't that what we sort of started talking about a minute ago? You describe a new species, you get it out there, now it has a name, now it can be conserved and blah, 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 right? Isn't that what we do? Well, instead, my scientific description of that species inadvertently facilitated its commercial exploitation. And just to fast forward, we now know about 14 there are about 14 known localities of the Lao newt. Depending on how you define a locality, you know, there's little sub-cluster lists. About 14 localities. What I will tell you is that every single one of those localities, local residents are harvesting the Lao newts for export into the pet trade. So anyway, I put out this little piece after this happened. I was horrified. And I found out, hey, you know, I talked to some colleagues and similar things had happened with a Rhode Island snake-necked turtle and some Goniurosaurus from southern China. In any case, we put out this little piece while I was a student here that just said, what are we supposed to do about this? Our jobs are to describe and publish, report on the world's biodiversity while we can, so that something can be done to conserve it, right? But what are we supposed to do if we can't publish on these, you know, such important elements of biodiversity like the Lao newt? Following up on this, this is kind of what happened. This was kind of the outcome. The outcome was when people would publish new species of newts and some of these other commercially valuable taxa, they would start to hide the locality, okay? So for example, like this guy. This is an Echinotriton, which is another Asian salamandrid. They have these really cool ribs, but highly desired in the pet trade. When they publish this new species, look at this. The type locality is under a rock on a mountain in southern China. Southern China is a big place. Look, it looks like this. So here, so on the right is their sort of, that's what they're saying the type locality is. Somewhere in that red zone is the type locality. I just happen to paste this over my state of North Carolina and South Carolina to show you how large of an area it is. Okay. The problem here is, yes, this might inhibit people from doing what they did to the Lao newt, right? But it isn't advancing science and it isn't advancing conservation. So it's sort of a knee-jerk solution to the problem. but I'm not sure it's the best problem. And this conversation is still going on. Same thing. And so this is now what is happening so frequently in Indo-Chinese and Chinese herpetology. When these Goniurosaurus geckos, man, they're heavily targeted in the pet trade, same thing. People just publish descriptions. We have no idea. They keep it a secret and so forth. Eventually, it probably will get out, but that's the way it is. This is not, it might in the short term stem the trafficking, but I'm not sure this is all what we think is the best for science or for conservation. But it's an unfortunate consequence of this kind of thing. Well, this didn't help. So the U.S. reptile and amphibian pet trade industry is worth about $1.5 billion. Okay. Reptiles Magazine is the biggest and most popular publication out of that industry. That didn't help. The Lao newt was the feature, new, beautiful, and soon to be popular. The magnificent Laotian warty newt was only recently discovered, blah, blah, blah. Buy your Laotian warty newt. That's tragic. Something had to be done. Something had to be done. And so it was mentioned in the introduction, I had support from the John D. and Catherine T. MacArthur Foundation while I was here as a student and also as a postdoc. And when we got that grant, the third grant in 2008, my desire was to find somebody in Laos to help with this problem. And this was really a pivotal moment in my career when I had the opportunity to meet Sompron Pimachak. At the time, she was just working in the fish production industry, in a fisheries department in Laos, but she was very interested in amphibians, didn't know very much about them. Laos, unfortunately, historically had no herpetologists or people with real expertise in amphibians. But through that Field Museum's MacArthur grant, this is important, through that Field Museum's MacArthur grant, we advertised a scholarship to support a student to enroll in the National University of Laos, brand new Masters of Science program in the biology department. And we interviewed a bunch of people. Sompron was clearly exceptional. We took her on for the job. She enrolled into that program. By the way, she became the first student ever to receive a master's degree from Laos, from the biology department in Laos. In any case, Sompron, her job as a master's student at the National University of Laos was to figure out what is the extent of this commercial trade in these things? What is the extent of the geographic distribution of these salamanders? And are there other conservation threats? And do we know anything about their natural history? Because so far, everything I'm just talking about is just the adults. We didn't know where the eggs or the larvae were or anything. And that's what she did. That's what she did. Months and months of tough field work, working in difficult and still to this day, a military sensitive area, not easy to work in. Conducting interviews with local people, talking to them. And what she basically learned was that historically, first of all, important, everybody who has a resource area within the range of Lao newts knows the species for the reasons I told you before. They're bright, colorful, they congregate in numbers, diurnal, and so forth. So everybody knew what we were talking about in these interviews. And yeah, they've always done this thing, and they would collect small numbers of them, dry them out, like you can see down there. Those are smoked over a fire, put them into whiskey and for medicinal purposes, but small numbers, just medicinal purposes, and probably not a real serious. These people had been living for thousands of years alongside this thing, and occasionally they would put some into some whiskey or whatever. Don't do that, by the way. But you know, okay. But the commercial exploitation, when foreigners started coming to their villages with pictures of this thing and saying, I'll pay you for it, and they can go out in just a few hours, scoop them all out and sell them by the kilogram, well, that's very different than what local people were doing already with these things. Anyway, Sompron, she conducted a mark recapture study on one particular stream. Here she is. She did belly pattern recognition for individual to recognize she had a little database going. So she knew she would do a little tail fin clip. So she knew that she'd previously captured the individual, saved the little tail clip as a DNA sample. It was great. It was great. She found the eggs. She found the larvae. She learned that they have Fs, which is an immature terrestrial stage that you see in this family of salamanders. She even found that occasionally, very occasionally, and these, by the way, are fully aquatic salamanders, it would very occasionally, the adults during a heavy rain would move over land from one stream to the next. She figured it out through pitfall trapping. She learned a lot. Skipping over these numbers, all I wanted to say was one of the most important things she learned, that says 10 localities, like I said, it's now up to about 14. But what she learned is that this is a species, one of those species, where it is not distributed throughout a stream, but it is distributed just at the headwaters of these particular streams where there are pools, and there they congregate. And when you are there, they are locally very abundant. You know that kind of species, right? So locally very abundant when you are right there. So, she came up with some pretty awesome numbers that were heartwarming to say, but across the landscape, not distributed that way. Very, very pocketed. And I mentioned this before. We learned from the interviews, just huge numbers of these. They would sell them not by the individual, but by the kilogram. What does that say? I mean, salamanders about a kilogram, right? It's a huge number, hundreds at a time. This is a little bit of a complicated slide, but just all I want to say is we did some species distribution modeling, Amanda Chunko and Sobran and I, and based on where we had ground truth, where we knew for sure the thing lived, we did some ecological niche modeling to come up with a climatic envelope about where it probably occurs. Forget the details here. All I want you to take from this is what we learned, at least from the ecological niche modeling and from the Sobran's all her extensive ground field work, is that this thing is probably and very truly endemic to Laos. The other thing we learned is those areas in gray are the protected areas of Laos. The species, there were no verified or likely to be localities within any of the existing protected areas of the country. Sompedon engaged in a very intensive education and awareness program, distributing local language posters in remote villages where these Lao newts, they had newts in their resource areas. These things were even hung in the Vientiane airport for a number of years. Trying to talk about this is important. Basically what this says is this is an important part of our natural heritage. I am found only in Laos. Please protect me. And even better, thank you to Sompedon. In 2008, she managed to get the Lao newt listed as the first amphibian to be protected by the country of Laos as a Category 1 listed species, meaning any commercial trade is banned. It didn't matter. The commercial trade was going on anyway, but at least she got it listed. The first, and still to this day, the only amphibian listed as fully protected in Laos. Finally, just 2 years ago, we finally got the Lao newt listed on CITES Appendix 2, which regulates international trade. And international trade is the major issue. It's long overdue. It had a lot to do with just very difficult issues within Laos. To underscore that point, in 2023, Laos was suspended from CITES for egregious trade violations of elephant and tiger and so forth. So it was not an easy thing to do, but we finally got it through. So even though Laos is not currently in CITES, at least this thing is listed on 2, and so it's still listed. Trade was continuing, and then there was a game changer. It wasn't looking good. And then some news came out that at first, and still is, I suppose, worrying news for global diversity of salamanders, but there's a really weird twist to it. And that is that in about, 2013, something like that, you're all familiar with amphibian chytrid fungus. This other thing, the salamander chytrid fungus was discovered in European populations of salamanders in the same family as the Lao newt. Of this other chytrid fungus, different, they described it as a salamander chytrid fungus. Anyway, it was being found in native fire salamander populations in Europe and so forth. And it was a real worry. What is this new salamander chytrid fungus, right? And it was causing die-offs in local salamanders. Well, look at this. Look at this. Through a number of laboratory experiments, what these researchers figured out is that the host reservoirs for this newly discovered salamander chytrid fungus are the Asian newts of Pachytriton, Paramesotriton, Synops. How'd you like that? Here, this just sort of emphasize the point. Those things in orange, those were demonstrated in the laboratory and through sequencing and a number of other, I think, rigorous methods demonstrated that is the host reservoir. So Asian newts are the host reservoirs that have evolved for, I don't know, millions of years or whatnot with this fungal pathogen. They're okay, they don't die from it. But guess what happens when you take Asian newts and they escape into the wild in Belgium? They pass on the fungal pathogen to the naive populations of the local salamandrids, and that's why the fire salamanders started dying. Now, everybody was panicking, and should. It's a serious issue. I mean, by the way, and you're thinking, okay, some newts are going around the world, whatever. Don't underestimate how big this trade is. Look at this. During a 5-year period, look in blue there, just looking at, and I can't just pick on Europe and Japan, so the United States is complicit in this whole. In our just 5 ports alone in the United States, during that 5-year period, we imported about 760,000 live Asian newts. Guess what? 99% of them were from Asia, and guess what? 91% of them were Paramesotriton and Synops, the host reservoir of this new salamander, chytrid fungus. And that's serious. That's serious. But when given a lemon, maybe we can make some lemonade here. And that is, look, and this is just a real quick plug for North Carolina. You know, we have more species of salamanders in North Carolina than any other state. We have 74 now, which I can't believe. By the way, 11 of those have been described since 2020. One of them by me, the one there at the bottom, but all 10 of those are in the genus Designatus. That's a whole nother talk in itself. But the point of it is that there's a lot of salamander diversity. They are the biomass of salamanders. If any of you worked in the Southern Appalachians, even if you weren't interested in salamanders, you were there to get snails or what other organism of interest or plant, you saw salamanders, didn't you? You cannot work in the Southern Appalachians and not see salamanders. They are a critical part of the ecosystem. And in any case, fortunately, B-sal, that's the name of that salamander chytrid fungus, has not yet been detected in the wild in the United States. And this doesn't work, that's not wood, but, and we hope it never is, right? But if it were to become introduced into, for example, the Southern Appalachians, into the Western North Carolina, it would have devastating consequences. I'm going to skip that. That was just to make a point that there's still lots of more undescribed diversity of salamanders in the Southern Appalachians. All right, given a lemon, can we make some lemonade here? And that is that this B-sal thing is serious. But you know what? It turns out Lautriton has it. And so if we can curb the import of Asian newts into the United States, into Europe, and so forth, for the purpose of preventing the introduction of this chytrid fungus into native populations, maybe this could benefit the Lao newt. And that's what this piece was. It was save our salamanders and do it by banning the live import of Asian salamanders. In 2016, the U.S. Fish and Wildlife Service moved a huge number of species of salamanders onto the Lacey Act as injurious. That basically just makes it difficult to bring them in live into the United States now. For a while, there was some interstate transport stuff that got overthrown in a federal court in 2017. In any case, there are at least 201 species of salamanders from around the world that are considered now to be potentially injurious species. So trade in bringing them alive in the United States is at least protected. Look what we did in North Carolina in 2018. I won't tell you who had a voice in this. We banned the possession of all Asian newts, live Asian newts in North Carolina. And some people originally thought, that's a little over the, no, it's not. We, again, are the salamander state. And all of these salamanders that we're bringing in alive for pets are the reservoir hosts of this deadly fungal pathogen. We cannot get that into the wild. In any case, just to kind of wrap up, I want to go back to Sompron, because this is really about her. What happened to Sompron after this? She did this wonderful thing. She got this thing protected in Laos. So she went on and did a PhD in Thailand. There aren't any PhD programs in Laos. And she worked on a different group of newts, Tylodotriton, okay? Same family, but sort of a distant relative within that family. Because while she was doing all that Lao triton, all that Lao newt field work, she was starting to find other newts. Well, when I first collected those specimens of Lao newts in 1999 and brought them back here to the Field Museum, those are the only newts known, only salamanders known from the country of Laos. By the end of Sompron's PhD, there are now 5 species of salamanders in Laos. So we've got 4 different species of Tylodotriton there now, those 2 at the top. She and I described together. The types of that one Nodialis are here at the Field Museum. Those are both endemic to Laos as well. So now 3 of the 5 known salamanders in the country, newts, are endemic to Laos. She did a wonderful job with her PhD. And by the way, I won't go into it, but that one that she, Podictes, that she named as a new species, as another subset of her thesis or dissertation, was a marker capture study, very similar to sort of what she did before. So we learned a heck of a lot about the natural history and ecology of a species at the same time we were describing it as new, which was pretty fun. Sompron today is a professor at the National University of Laos. And she has students of her own, undergraduate and master's students, who are studying amphibians and reptiles. And there is no greater joy in my career than having seen that. That's Sompron. We've helped through that MacArthur Foundation grant that came through the Field Museum and other funds. We've helped to develop a collection, a herpetology collection at the National University of Laos in biology. You can see, well, it's a little bit cut off, but there's an NUL as their acronym, NUL, and it's okay. It's a pretty nice collection. It's an air-conditioned room. We've got good jars, good quality ethanol. Air conditioning goes out, a lot of power outages. She's blocked out the windows. They do a pretty good job. She's got a nice little database going. They have a -20 freezer with tissues. They're doing a pretty good job. They're doing a pretty good job. And I just want to say, this experience that I had, I started out again. Remember how the story began? I was a graduate student. Well, this is even before I was a graduate student. I was working alone. I had no fellow herpetologists in Laos and so forth. I made this discovery. I opened up a very unexpected can of worms. The problem just kept snowballing and snowballing and snowballing. And the solution to it was to find a Lao national who had expertise and interest in this problem. And so having that experience completely changed the course of my career. And the Field Museum has much to thank for that. In 2017, we got a grant. This was a pretty neat thing. It's called a Peer Science Grant. I don't know if any of you have ever had one of these things. It's out of USAID, but they give you money that partners up with an existing National Science Foundation grant. But anyway, we got a grant, and it was called Conservation in the Lower Mekong, Empowering Female Herpetologists Through Capacity Building and Regional Networking. What we brought were 6 herpetologists from Laos, from Thailand, and from Vietnam to the United States. Much of their time was spent here at the Field Museum in 2017. They each had a little project to work on specimens from their home country, measurements to make, scales to count, et cetera, while they were here. But they also benefited greatly from interacting with many of you. They got to tour your collections and so forth. There's John Bates down there. There's Larry Heaney showing off his collections. There's Kevin Feldheim showing the Pritzker DNA lab. There they are in the upper right. There we were talking about frog taxonomy with Bob Inger. What a wonderful, wonderful experience that was. And later that year, Sopotuan came to Field Museum on some Fulbright money. And we went through specimens like I'd never have before. And she had the opportunity to meet Bob Inger and of course Tan Phui Lian, to see all these early collections that I had made in Laos long before she was ever on the scene. And wow, did she benefit from that experience. And I'll just say, I'm gonna put my neck out there and get myself in trouble. You see that frog picture that's on the wall right above Bob Inger's head? That's this one, real quick. This is a species that Bob actually described from Laos. Maurice Cottelot's an ichthyologist, and he got some of these Amelops frogs and some ichthyological collecting bycatch, sent them here to the Field Museum, and Bob and Maurice described as a new species, Amelops cremnibatus, type localities in Laos, not too far from the Vietnam border. See that beautiful drawing? Figure 1 of the holotype, that was drawn by Tan Phui Lian Inger. And that's the picture that was above Bob's head in that earth, was of a Lao frog. And that's why we took that picture right there. There's more to it than this. Here's where I'm a dead man. But last year we did a project and we looked at morphological variation, mitochondrial and nuclear DNA variation within Amelops cremnibatus across its range. It mostly occurs only in Laos. It's a wee bit into Thailand to the west and a wee bit into Vietnam on the east. But in any case, we partitioned it up into 4 species, cremnibatus and 4 new ones. And one of them we named after Tan Phui Lian. And I just want to tell you, I won't read the whole etymology, but part of it is, in addition to that beautiful holotype, what not beautiful, figure 1, I love that. Phui Lian's scientific contributions and her generous hospitality to us and innumerable visitors to the Field Museum of Natural History have had immeasurable positive impacts on Southeast Asian herpetology. So that was a real honor to be able to name that after her. I'm going to close up. I see I'm running out of time. Just a couple of slides. Where are we going from here? We just got a grant. Dave Wiesrock is the PI. He's at the University of Kentucky. A wonderful salamander phylogeneticist and a wonderful person. In fact, I met him for the first time when I was a graduate student here. I met him at TGIF. This was like probably 2003 or something. Yeah, there's real benefit to that. You don't think when you meet somebody up at the happy hour that 20 years later we could have a grant together. Anyway, he's a wonderful phylogeneticist, mostly in salamanders. We got this grant from Revive and Restores Wild Genomes Amphibians, and what this is doing, I just want to point out, so Dave Wiesrock's the PI. 2 of my Lao collaborators are on there, obviously Sopotuan. I just want to point out Jeff Frederick. Some of you might know him. He's a finishing postdoc with Sarah Ruan, and he just, as of last week, joined on as Dave Wiesrock's postdoc at the University of Kentucky to help move this forward. But in any case, what we're doing is we were able to get a zoo captive individual. We were able to generate a really high quality transcriptome from it by taking, you have to sample, brain and spleen and lung and liver, all these tissues. We were lucky. The Mississippi River Aquarium and Zoo in Dubuque, Iowa, actually had a captive colony of Lao triton. And they were so horrified to learn of the history of the Lao newts that they were keen to at least let one of their illegally captured and exported and purchased animals be part of a positive conservation effort. So they donated one of those animals from Dubuque, Iowa to this. We've generated that transcriptome. And now what we're doing is developing a custom bait kit that will basically let us do genome-wide sequencing of about 100 individuals from all of those about 14 localities. And what our hope is, and we have some preliminary RAD-seq data, for those of you who do this, you know what I'm talking about. There is some genetic structure. There is some genetic variation that correlates to geography across that, albeit small, geographic range of the Lao newt. And the idea is that if we can get some genome-wide variation, maybe, if we're lucky, we can narrow down just the few markers that one might need, where we could design some primers that could be amplified and sequenced with a Sanger sequencer in Laos that might be able to determine the origin of that individual salamander when it is confiscated from trade, and then potentially either repatriated back to that part of its geographic range or perhaps being put into a responsible breeding program or whatnot. And so the idea is, we don't know yet if this is going to work, but we're hoping we're going to come out of this with a primer set, a Sanger sequencing primer kit that can be used by Sompuran and Sangvilai and their students in their brand new molecular lab at the National University of Laos. That's no Pritzker lab, but it's not too bad, right? They can do some basic stuff. They have to send their Sanger sequencing out to Korea, but they can do extraction, they can do PCR, et cetera, et cetera. And so this is what we're hoping for. This is our new challenge in Laos at Sompuran. That's maybe the biggest smile I've ever seen on Sompuran's face in that photo. And this is the end. I want to remind you of why I have a sticker up that says, "What would Field Museum do?" What I gained from my time and continue to gain from my time at the Field Museum continues to have a tremendous impact on my career, but not just my career, on my students' careers, and now her students' careers. So I want to say thank you very much for what you do. It matters. Thank you.
Bryan, thank you so much. That was so engaging, very passionate, like a lot of sadness, happiness. Thank you so much once again. And we have some time for questions.
Hi, Bryan. Update on their status. Are they still extant in the wild? Can they still be found or protected or?
Yeah, good question. I should have mentioned that. So they're globally listed as endangered. They almost got listed as critically endangered, but just of a small fluke. Anyway, they're globally listed as endangered. Well, we think they're still extant at least at some of the sublocalities of all of those 14 localities. So we still think there are some in the wild, but it's been several years since we've been able to do repeat, COVID shut everything down and so forth. So I'm hoping that what happened in recent years, certainly the CITES listing a couple of years ago, but especially this BSAL and the United States, Europe, and so forth, cracking down on live imports of Asian newts, has helped to stem the flow, but we don't really know for sure. Sompuran's been back to some of these places. They're still there, but there's still demand. The demand has not stopped. A lot of it now is going into China and into Thailand. But again, where they go from there, I don't know. Thank you.
I have a question, Bryan. Also, I wanted to mention that I've always been very proud to be in the Field Museum, but I feel even more privileged and very happy to hear your story and learn a lot more about the history of the collection. Has there been any captive breeding for the Lao newt?
Yeah, it's a very good question, a very interesting one too. So there has been successful breeding of the Lao newt, primarily in Europe. But the issue has been, they have been able to reproduce them, to breed them, because they do pretty well. Like I was mentioning, they do pretty well when they're in captivity. But what they have learned is that they'll breed well, they'll hatch well, they'll metamorphose into the terrestrial Fs, and then they die. They can't seem to get those little tiny terrestrial Fs to survive. Last I heard, that was always the bottleneck. And so there have been, I'm always surreptitiously watching and seeing what's going on with the internet. You can find, I'll occasionally see a captive bred individual for sale in Europe, huge dollars, especially if it's post-F'd. Usually they're sold when they're Fs, right? Because that's when they die, right? And so in some ways it's a little bit of a masquerade, right? Because it's sort of like, and I have people say to me, "What's your problem? We can breed them." I've got 100 of them for sale right now. 100 captive bred ones for sale right now, but they're all Fs and they won't live, at least not yet. So it's a facade.
Bryan, thanks for this. How long do the adults live, do you think?
It's a good question. We don't know. it'd probably be possible to do skeletal chronology. Unfortunately, due to someone's limited budget and resources, none of those individuals were permanently marked. I wish if I could, we all want to do something differently, going back in time. And I wish we had marked with like elastomer dye primers or something like that. So it was like a permanent mark, because remember how she was doing the little fin clip to make sure? Well, she kept a couple in captivity and fin clipped them in order to find out how long it took for that fin clip to grow back. And it grows back in about a little over a year. So unfortunately, we can't go back. Now, of course, she has those belly pattern, she still has that database, but it would be, I don't know. But it would be probably very interesting to go back to that same little 4.7 kilometer stretch of stream that she did her master's thesis work in for that marker capture study, and to see if there's any way to possibly recognize any of the same individuals. Otherwise, I guess somebody could do skeletal chronology or whatever. Anyway, we don't know.
Sorry, one more question. So has anything changed with the protected status of the lands in that area?
No, there's still no protected area within the range. Soberon's been working very hard on getting a provincial protected area, which is a lot easier than getting a national protected area. Province, of course, being like a state, so a more local level, and been tough. But here's the funny thing about it. All those species, remember that collage, all those amphibians, all of those things, the problem was this rapid rate of deforestation, right, and so forth. That Lao knew it does fine without the forest. Did you remember the pictures of the type locality? I mean, denuded, heavily bombed, cattle, I didn't point it out, there are cattle grazing in the background there at that type locality. They're pretty resilient. I mean, we find them in those headwater streams with and without forest along the borders of the streams. They're pretty tough. And they were doing okay until, darn it, people decided they want them as pets. And so I do worry about the deforestation. I mean, it's serious. I don't mean to undermine it. But in the case of this one species, the deforestation is not the primary problem. It's rare to be able to say that, but it is. It's the overexploitation for the pet trade.
I think we have time for one more question, if there is any.
I can talk loud. I'm a college professor. Is that okay? Can people hear me?
Is that okay, Todd?
In these second generation breeding, like they always die in the end, do any of these breeders that you apparently had contact with, have they ever gone like, what would be a population, a colony, as opposed to a one-off breeding event?
Yeah, okay, so this is a very good question. There have been some F2s. I've seen that, because there's been a number of articles published in sort of the hobbyist literature in Europe. So there's actually a tremendous irony, and I hesitated to go into this, but the person who has had the most success in breeding the species and has published most of the hobbyist literature on how to breed them was one of the first people to go in there with that Journal of Herpetology paper, and who actually kicked off the demand for it in Germany. So it's a tough one. I can leave I'll leave it to your imagination. My relationship with him isn't very good. And so but what I can see is they've had some success. So I think they're figuring some stuff out. But what I can tell you with certainty is they don't have it all figured out, because it's not common. Usually by this point, after so many years of demand, if these things could be captive bred in large numbers and so forth, they would have swamped the market by now. And it's not the case at all.
Well, once again, thank you very much. And thanks everyone for coming to the talk, for such a great turnout. Thank you.
Thank you.